I'm not sure if you
Okay,
can see the
great now.
the screen or not. So I'm sharing. I can
Yeah.
send you this,
No, I
John.
can see it. I can see it. I can see it. No
So you
problem.
see, we have Medera. Celadon is it doesn't exist anymore. It was an old company, and Roger licensed the technology to this company as well. And then we have Aspire, right? So basically, everyone. So the three companies, and there are actually several other existing companies, and many in the past have also pursued the calcium pathway, calcium handling pathway. I mean, as I told you before. So Tenea, AskBio, they are all coming after this calcium pathway with the circuit 2A
Look at two. Yeah.
protein being the ultimate effector protein. So this is an important pathway in basically all forms of heart failure and cardiomyopathy, and circuit is universally downregulated, right? And circuit is an endogenous human protein found in the human hearts. So
Mm.
hold on one second. Sorry about this. So I have to. Okay. Okay. Um, yeah, so this is the common pathway that many companies are working on. I mean, here AstraZeneca has also been working on small molecules. And what is that? I showed you two other related trials, right? I mean, they were.
Yeah.
I mean, the small molecule was also they're trying to target.
So the one that was pretty good, like 17%, was it Tanea?
No, not Tenea. That was a Tectonic.
Oh, they're not in this
a
list, right?
a separate company. So that was more like for more specifically for pulmonary hypertension, uh,
Oh, okay. I see. I see.
have pulmonary hypertension patients, which is a subgroup, a patient subpopulation subset of what we look at. So
Okay.
and for these companies, Celadon and also AskBio didn't have access to the mini heart technology, right? So for us, I mean, as you know quite well, we have the mini
Yeah.
heart hardware and software based automation, and that's been exclusively available to us. So we use it to optimize dosages, to discover
Mm.
the targets in collaboration with AstraZeneca. So that's a key difference. I mean, as you know, our mini heart technology platform has generated data that led to FDA clearances for the IND and also fast track designation. And more specifically, that's for HFpEF. Um,
Could I clarify this, right? So
yeah,
AstraZeneca is also testing their small molecule on your mini hub platform. So
yeah.
technically, in this list, only Medera and AstraZeneca has access to this mini hub. The rest are flying blind, right, when they're dosing.
Exactly. Basically.
Okay.
So AstraZeneca has access to part of the platform, and they are still screening, right? So and I think I told you that we have installed the part of the mini heart system in their R&D center in Sweden, Gothenburg.
Yeah.
Yeah.
Yes.
So
And
all yeah.
do they have a small molecule in play already in the clinic?
They have had been developing looking at several small molecule candidates,
Hmm. Okay, that's fine. Yeah.
but pretty early stage. Yeah.
Okay.
So all three companies have used the intracoronary delivery protocol. I
Yeah.
mean, you know that Bayer is injecting manually, and Medera is using a pump to regulate the blood flow.
Yeah.
And AAV1, AAV1, AAV2I8, we talked about that last night as well.
Yeah.
So we are directly injecting into the heart the serotype, whether it's AAV1, AAV6, AAV9, AAV2I8, it doesn't really matter. The
Yeah.
uptakes been shown to be very similar. And for Celadon, when they were doing it about 15 years ago, right? 15 or even longer than that. So
2007.
yeah, so
Um,
almost like 20 years now. So little was known about AV, and AV9 was not even found or known
at the
back
time.
then. So
Yeah.
that's the history, right? And for
Sorry,
formulating,
Celadon. Just to understand, Celadon is part of us or what?
no, that the company doesn't exist anymore. I mean, you asked about Cupid 1, Cupid 2, and the American
Oh, yes, yes,
Association.
that's
So
right, that's
they
right.
did the Cupid studies, right? So they licensed the technology from Roger. So, but
Oh,
what
right.
I'm trying to see is that that was a long time ago, 15 or almost 20 years ago. So, how are things different now? The formulation is different. The manufacturing method is also different. So, meaning that now we have less empty capsids,
Yeah.
and less broken DNA, and more consistency, and things like that. So, the purity, so to speak, and potency of the manufactured batch is very different from what they were using, almost like 20 years ago.
Got it. Got it. And we're now only talking about intracoronary, right?
Only
Is that
intracoronary, not even IV, right? So
okay?
intracoronary.
And
So
well, I just want to be sure, right? This is currently both of you assess or the data shows is the most superior method of doing this, right?
exactly, exactly. So I can send you this table, right? So
Okay.
just want to highlight to you some of the differences, and so that when you look at some of those other comments or critiques, and you understand what
Ah,
we're
yes.
doing and why we feel that we are more superior, right? So for Celadon, they were using dosages that were anything between 3 to 30 times smaller than what we are using now back then.
Mm.
So lower dosages, meaning that's the other, within the other side of the dose response curve. So as a result, they had only a 0.5% transduction efficiency in human biopsy. All right.
Oh,
So we
okay.
have higher dosages, and Bayer is also using higher dosages. And Roger and I are gonna tell you more what is associated with these dosages. But
Yes.
basically, with the higher dosages now, we can get like about almost like 100% transduction efficiency in humans, and also
And
in
this is in the mini heart model, right?
mini heart, and also different animal models as well. So
Right.
back then, basically NIH capped
Yeah.
the
Yeah.
dosage. They could
Yeah.
only go as high as 10 to the 13. So that was a very
Mm.
different time, not only in cardiac but also in gene therapy in general.
Mm. I have a curious question, right? So we are later generation than Bayer or AskBio. Why are we stuck with the same dose? So if you
No,
scroll
it's
up,
just
right?
that you know what I can show you the dose response curve. It's just a very steep. It rises very steeply. So that's why you have to look at the most dynamic part or steepest part of the curve. So
All right, I see.
half a lot of concentration is what it takes to make a difference. But when you go too high, then you have to start worrying about immune rejection, or T cell responses and things like that.
I see. I see. Okay, but considering if we are both at the same dose, why do the both of you think that we are superior to AskBio? Actually, that's the most important question, right? And where
Yeah.
are they in that process? Have they flunked, or are they proceeding with phase three? Because I saw a number like 150 patients, right? So just trying to the most important question today when we go into Morgan Stanley is so that I have some base understanding, right? That they are not going to be the standard of care when we actually go into trial. Yeah.
Yeah. So, I mean, I think, first of all, the, um, sorry, the AskBio is not doing half-path.
Oh
And
yeah. Okay.
yeah,
Yeah, that's
so
important.
we have the
Yeah.
big advantage of being doing the half-path. And also in their half-path program, they only do non-ischemic cardiomyopathy. And I think
Hmm.
that, in terms of comparators, um, um, where can I up? Oh yeah, here, share. Okay.
Oh
Uh,
yeah. Hold on. I'm just gonna allow it. Yeah, of course. 1 minute. This is sorry. I'm the host. Yeah. Please do. Yeah.
yeah. So
Mm. So maybe while you're looking for the document, right, I just explain the question here. Um, People are unclear where AskBio. In fact, I don't think our research can show right where they are, how committed they are to their program. If so, this is double-edged sword, right? If they're going very well, then we're going to be second class. That's the scare, right? Of course, now Professor just explained that no, they're not going after half path, right? But if they're not going well, that's the other problem. Do we have a commonality that is like going to
mm,
set us back in the trial? So
Yeah.
I'm sorry, this is like a too good or too bad. It's not a good outcome, right?
Yeah, I
For
mean,
both companies.
I think bad would be bad for the whole field. I don't think it's, but I think there are
Okay.
certain tweaks that we can point to that we are different and
Mm,
that can set us apart. So the target for AskBio is shown here basically. Um, it's called I1C, and it's an indirect way of activating circuit 2A.
uh,
This
yeah.
is circa pump. There's another protein called PLN, phospholamban, which in its unphosphorylated form inhibits circuit 2A. And upstream of that, there's a protein phosphatase 1, which in heart failure is elevated. And it keeps phospholamban unphosphorylated because phosphatase dephosphorylates. Uh,
Sorry to interrupt. I'm not really seeing the slide, or is it just me, or
Oh no, I could see the slide.
you can, Johnson?
Yeah,
Okay,
yeah, I can.
that's fine. That's fine. Don't worry about it. I know the
Okay,
slides. It's-
yeah.
yeah. So I1C is this constitutively active form of I1, which is endogenous, but constitutively active form is not endogenous. It's a synthetic peptide that has a mutation in it and it's truncated. So the body uh, thinks it's, excuse me, foreign. So that's going to be important in a second. So what it does is that it inhibits um, protein phosphatase 1, which is elevated.
Mm,
Protein phosphatase 1 inhibition
downregulated. Yeah.
phosphorylates phospholamban, and circuit 2A can now increase its activity.
All right. It's an indirect way. I see.
And so, two indirect ways of doing it. Okay.
Yeah.
Um, Fast forward, the 2I8 I1C trial is the following. It has two uh, cohorts. One is 3E13, which is similar to us, and the
Mm,
second cohort is 1E14 uh, intracoronary delivery. And so it's a little bit higher. It's higher than our high dose.
yeah.
So and when you look at the data, this is their uh, table of data for the low dose and high dose. And in general, I mean, it looks very similar to ours. In fact, the high dose doesn't seem to be doing so well in their
Mm,
as well as the low dose. So the low dose is the one that they focus on. However, one one area that is very different than ours is that the T cell responds to the capsid and the transgene. So remember, the transgene, the I1C that they have, is a synthetic peptide, and the 2IA
mm,
is a capsid that they've manufactured themselves. So it's also not
I
uh,
see. That was what Roger was telling me last night. Yeah. Ronald was telling me last night, right? Yeah. Yeah.
yeah. So what they found is that the when they looked at the T cell response in the two doses, two cohorts,
Yeah.
um, both in the low dose and in the high dose, they had T cell responses to the 2I8 capsid. So you can do a specific capsid response.
All right.
And everything in red is high numbers. And they saw it both in the low and the high.
But
The
the low
first
one is a lot higher, right? Or am I reading it wrongly?
Yeah, I mean it's both of them are high, unfortunately. Yeah, they were,
Oh,
the
okay.
first three patients in the low dose group were done during COVID, so they weren't able to get the samples. But
All right.
yeah, but you see that there's, now, okay, I mean the 2I8 capsid you can sometimes get it, but more concerning is the fact that at the I1C, so there was a T cell response to the actual transgene.
Mm,
This is what's shown here. both at low dose and high dose.
I
So
see.
there was a T cell response to the capsid, T cell response to the I1C, and this is sort of two representative patients where they have high uh, T cell response to the 2I8 capsid, high response to the I1C. It was
Mm.
uh, they also had an increase in LFTs. So ALT and AST are markers of liver function test in both these patients. And
Oh, I see.
yeah,
Yeah.
so yeah, so the straight lines are the ALT, AST, and the
But
bars.
they weren't sustained, right? They were
No,
transient,
no, they
right?
were not sustained. Both of them were not sustained, but still, I mean, I think it just shows that you have a T cell response, which can uh, mitigate uh, your biological response because you're
All
trying
right.
to get rid of
Yes.
yeah.
Yes. I
Um,
see.
so
Actually,
basically, they
Professor, do you
found
mind? Yeah, do you mind going back 3 slides? I just want to take a look at the trial design, just to compare the hours. Ah,
and
yeah,
I'll
this
show
one.
you ours. I'll show you ours.
Yeah, okay, okay. That's helpful now. Yeah, I'm just looking at the numbers. Okay, that's helpful.
Yeah. So ours, it's both 3E13 and 4.5E for 13. So Ron, I added some new data that I just got today in terms of the
Oh, wow.
higher
Yeah.
dose. And they look, overall pretty good. I mean, we don't have 12 months data in many of them, but we have in some of them we do. But again, I think it looks, very promising that the higher dose may be even better than the lower dose. But the lower dose also is quite good, as you can see. So there's not
Oh,
much difference.
wow. The 1023 guy had like a 720 in the 6 months walk time.
Yeah, I mean, 6 minute one, and look at how his NT-proBNP, it went from
Oh,
5.5
yeah.
to 47. His EF went up by 10%. So I mean, there's clearly some great numbers here, and
And this is only half the cohort, right? I remember the high dose also five guys, right?
yeah, this is the 4.5 E13.
But there's supposed to be five of them, not three, right?
No, no, there's three of them. Five is the HFpEF. You're thinking of HFpEF.
Oh, okay, okay. I see. Okay,
Yeah,
got it, got
HFpEF
it.
has five and five.
Okay, I see. Okay, so even the half ref data is still being updated right now, right?
Yeah, yeah. I mean, I think it was updated today, and the HFpEF hopefully will have some more data coming up. But it looks all pretty good in
I see.
general.
And
Um,
and yeah, so if we were to pull back, right, you're saying the T cell response plus the safety issue is hamstringing the whole AskBio program. Is,
yeah, I
is
mean,
that
look, it's not hamstringing it because they continue to use it. This is the uh, intracoronary delivery in patients where you see the low dose, a little bit
mm.
higher dose. This is what Nanocore did,
Oh,
and
the first two is ours, right? Oh, no, Celadon and Celador, yeah, are both there. Yeah.
yeah, Celadon, Sardacore, and then the S-Bio, they're all intracoronary. This is just to show that we don't use steroids, that in general it's much safer than
Yeah.
the rocket ones where they do intravenous, much higher doses.
The rocket guys like muddied our research. We can't tell what's going on, right? I see.
Well,
I
it's
see.
yeah, it's just very high doses. I mean, it's just they get T cell responses, they got immune responses are extremely high, and that really is a
Yeah.
problem.
And in this chart, right, the us bio guys look decent. Is that right, or am I reading it wrongly?
Yeah, I mean, I think the nano, I mean, they had positive T cell response. They had no SAEs.
Yeah.
Uh, the other trial, the Gene Fit trial, which is their phase two, I mean, it's blinded, so you can't know who's gotten what. But so far,
Yeah.
they've had no stoppages for SAEs or anything like that. So I think for us, as a, for starter core, it's all positive because, it validates the intracoronary, it validates that, now even AskBio bears used it and they've had no
Mm,
SAEs at lower doses, while Rocket continues to be mired in like hell with their
yeah.
death, capillary leak syndrome, TMA. I mean, they've
Oh,
had
this,
to.
this is an awesome slide. I mean, Ronald should have it in his deck. This is important. Yeah, okay. And if you were to help me summarize, right, what is the problem with AskBio or Bayer? Is it a lack of efficacy because of the T cell negating it, or is it a safety issue, or actually they don't have one? They might still succeed in HFrEF, right?
I mean, I think, look, they may succeed in HFrEF. We don't know. I mean, because the phase two is going to read out in end of 2026,
Six. Yeah,
early 2027.
yeah.
So there's still more than a year for the final readout of their phase two. But
Mm,
that's the only thing they have is a phase two, the I1C trial in non-ischemic
mm,
cardiomyopathy. And I've listed here for you all the differences that, we have in terms of Circa versus AskBio. But I think these are just to highlight the differences.
yeah.
But overall, if they come out, let's say, positive in their HFrEF, it's not going to affect us for us because
Yes, yes.
our
This is
HFpEF
context.
and DMD cardiomyopathy programs are really the marquee programs for us. But it validates our methodology.
Maybe a different question, right? Why didn't Aspire dare to go into HFpEF?
Uh, because there's much less data on I1C in HFpEF than there is in with Circa, and probably Circa is much more powerful at dealing with HFpEF in
I
terms of
see.
relaxation, because as opposed to I1C.
So we are
Uh,
doing something like no one else has done in HFpEF, right? So
Absolutely. Yeah. It's
oh, okay, I see.
first in human.
Sorry, because I this is confusing to me because generally the guys in Hong Kong, right, or we look at oncology a lot. We're very familiar with it, but when it comes to cardiovascular, it's a bit confusing the landscape, right? So I didn't know whether we are fast follower or me better, right? In this case, we're not, right? We're testing all the hypotheses ourselves too, right?
Absolutely. Absolutely. So this is
Mm,
first, for the for the HFrEF program, I mean, we have a legacy with Celadon, but that we're continuing and improving on. But for HFpEF and DMD cardiomyopathy, we're first in human. I mean,
okay.
concept-wise, uh, everything else, it's the trial design is extremely
Yeah.
uh, specific for HFrEF. No one has ever done anything like this in terms of the trial design. But everyone, loves it because it's just such a unique. And as I said, it was like a wall-to-wall. The session had like standing room only, and
Mm.
it was very well received.
Okay. Then one more question, right? Because we're trying to pivot or the entire company are tilted with a lever, right? So in that sense, we are trying to get to a phase three trial or even fund the whole phase three trial. One of the criteria is the FDA doesn't get too demanding in terms of the number of patients, right? So one of the flags that came out during our DD was at the Aspire trial. I think the phase three was 150 guys, or even or I don't know whether it's two arms, right? That could be an issue as well. And then later I could send you more about Palnova, right, which is trying to do pulmonary hypertension, right? But by via ablation, and they're claiming to have an effect on HFpEF patients too, right? A huge beneficiary effect. I get it that they are not competitors; they're probably complementary. But if their trial is being asked of 500 people, right, negotiated down from 750, right? I don't know whether single arm or I think it's dual arm, right? Does that impact on us? As in, we probably wouldn't be able to get like a 70, 80% request through, right? For a phase three trial.
Yeah, so for the half ref, I mean, it'll probably go, we're doing right now a 50 patient two-arm study, 25 and 25. Uh, Whereas AskBio is doing 150 patients, but two doses versus placebo. Now they're doing two doses because they saw that the higher dose maybe didn't do as well as the low dose, so they wanted to
Hmm.
hedge their bets with the higher dose. Um,
All right.
so the yeah, so
I see. And so do you have an inkling that what would the because IQVIA has given the quote at 80 million US, right, for two phase three programs, including the DMDCM, right? Um, what does that entail in your discussion with the FDA? In a half path phase three, right? Is it going to be a single arm or no chance of that? Or if it's a two arm trial, what's the numbers required, right? Based on your effect size now.
yeah, I think that if you look at the effect size for the HFpEF program, it's relatively strong. It's 25 to 30% in terms of the peak wedge pressure reduction. So we
Yeah,
expect the FDA to allow us to do maybe a size of about 100 patients for phase 2/3, so a registrational trial, about 100, maybe a little bit more, but
yeah,
not more than that, because
two arm.
if you
Yeah,
have that kind of a response, you don't need that big of a sample size.
I see. Is it a two arm trial or they allow you to use a model placebo?
It'll be no two-arm trial. I mean, the one-arm placebo, one-arm open is really for rare diseases. So it may be for the
Uh,
DMD cardiomyopathy. So definitely.
I see. And Ronald, you fully agree with that, or there's something to supplement to that?
Yeah, I mean, this is what we have to find out, right? So from the FDA, I mean, that's been always part of the plan the whole time, you know. But we can always, I mean, strategically, our chief regulatory officer has been saying, I mean, it doesn't mean that we cannot push the limit to,
Hmm,
to go with a smaller size. But
I see.
that's been the initial
At
plan at
risk.
the time. At the end of the day, it all depends on the entire data set. What we can say at this point is that look at cohort A, it's very promising. And cohort B, the higher dose, it's not like AskBio. They've been more limited by the T cell responses as a result of the capsid and also the engineered peptide. And we don't have the
Yeah,
same limitation. And early efficacy is indicating that the high dose functions even better than the low dose, right? So,
hmm,
and even with the low dose, the difference is already substantial enough at about 30%, right? So you know what I
yeah,
mean? So if we see a even better response with the high dose, about 30%, so
yeah.
logically, the bigger the difference, the smaller the sample size is going
The
to be.
size.
We're pretty optimistic because 30% is a big number, a big difference, right?
Yeah.
So,
The Could you help me out a bit, right? Because this is exactly that slide in your deck where one of the middle read, the middle bar was 17 -17%, right? You tell I didn't quite understand the. Could you flip to that slide in your deck
that's the
to?
20 watt. So it's not the peak exercise.
I see. So,
So peak exercise meaning that you're stressing the heart even more to amplify any differences, right? That we are seeing.
all right.
So
Oh, so
maybe we can take that out in the deck. Scientifically, I think people want to see that there is a dose response. You know what I mean? The stronger the exercise, the bigger the difference that you see. But in this
yes.
case, you want to show just two extreme cases, at rest,
All right. I
just
see. I see. So
peak
so
exercise.
if you're not stressing the heart too much, you're not moving that much away from the
Exactly.
baseline.
Exactly.
20 and the 20. Sorry, I'm not familiar with the trial, but what does 20 watt mean? The amount of
20 watts
stress.
mean like a moderate amount of exercise. I mean, that's usually somewhere in between. But again, I think the rest and the peak exercise, especially peak exercise, is the one that really um, uh, correlates with mortality.
I see.
So the
That means
higher your peak exercise wedge, your wedge pressure
Mm,
at peak exercise, the higher your mortality.
I see. And that minus 20%, right, is the established benchmark, right, for clinical meaningfulness. Is that correct?
Yeah, I mean, it depends on the parameter, but yeah, about 20% is the benchmark.
Yeah. So if our high dose comes in substantially higher for the peak exercise, peak wedge part, right?
Mhm.
Then
Yeah,
higher than the low dose, then you're likely to progress only one dose into a phase 3, and that will be the high dose, right? That's where we need only 100 patients. Did I get this last summary line correct?
exactly. I mean, even at the dose that we have right now, what we saw with the in the HFpEF trial is that at that low dose, we were
It's already
able
worked.
to, it's already bringing it down nicely. So, I don't think that we had to do much more than that. So, yeah.
Yeah. When is the FDA meeting? In Ronald's timetable, right? There are two sets of meetings. I'm sorry, I don't know where my folder is, but where do we get an answer or initial indication of the phase 3 trial design? Yeah.
Yeah, I mean, I think that it's going to be an RMAT application, hopefully by the end of this year, plus an FDA request meeting. We're waiting for a few points at the 6 and 12 months for some of the high dose of the HFpEF. But hopefully by, in Q1 2026, we'll have the FDA meeting.
Okay. I'm just looking at this this file here. Oh, you can see mine, right? Oh yeah, yeah. I think Ronald's showing.
Yeah,
I
Johnson, if
have
you see this slide,
yeah.
take a look at this. So this is another trial. So see the 20% cutoff. So TX45 is by tectonic. So this is
Mm.
logic that we talked about yesterday, right?
Oh, they
So
are quite good then. Yeah,
they did 20 patients, 19 patients in total,
yeah.
uh, all doses. So this is half path, and in the US, it's only half ref and half path. There's no,
Mm.
you know. So, but basically, you see that they did only 20 patients, and they managed to use this data. This is phase 1B study. I think they raised like $150 million just based on this.
All right.
You
And
can take a look. I mean,
this is peak, right?
Tectonic is a public company now. So I think 150 million or something like this, something in that range. So even with the low dose, we are already seeing this is 32% because they're only doing only looked at rest. You see.
Oh,
So even with this, they managed to raise like 150 million dollars or something in that range. You can look at the
no,
exact.
in the US is different, right? I think Asia the margin of error is needed bigger. But just so I understand, this is the peak data, the peak
Well,
difference
we're
or
showing the rest because they did only at rest. They didn't even do
oh right.
peak. Yeah. So that's what they
I
did.
see.
So that's why we're showing, I'm showing the same data set collected at rest, but not the peak.
Got it. Got it. Okay, got it. And are they progressing or have they talked to the FDA?
Uh, Roger, do you know?
Yeah.
I don't know actually there what they've.
Yeah. So
Mm.
It's
this
not public. I mean, it's not publicly cited.
Announce yet.
another
Okay.
another this is another study. I think no, sorry, I have to correct this. This is
Yes,
exercise
yes.
in 29%. So this is the rosamy. So you see, small molecule and compared to ours.
I see. I see.
So
Okay.
performed actually also by UT Southwestern for this study. So the same site.
All right. All right. Okay. No, no, this is now I'm a lot clearer about why the excitement by Kings on all this, right? Yeah. Okay. It's perfect
But
then.
once again, I mean, for us bio, keep in mind that HFrEF and HFpEF are different. The
Yes.
patient populations are different. Admission
Yeah.
criteria are different. The endpoints are also different.
Mm.
So it's like
And
comparing
there's
apples and oranges. You can kind of you can say that, right? The only
yes.
difference is that we do also have a HFrEF program, But as Roger said, our marquee programs really are HFpEF and DMD cardiomyopathy, and both are first in human. So they are not
Mm.
really compete for these two programs. They are not competing with AskBio at all.
Mm, mm. This is perfect then. Yeah, so I guess I only have like that. There's a Type C meeting, right, for GMP drug production. That's for production, right? But the Type B will be the clinical design, right, for phase 2B/3, and that's due in 2Q26. That's a correct summary, right? We will know how much is needed in 2Q26,
Yeah,
right?
exactly.
Hmm. Okay, no, that's extremely helpful. I think we covered all the ground today. Yeah. So is there anything else I missed out or yeah.
No, we can. I mean, Roger and I can send you the file so you have a record of that, right? And feel free to let me know and let me
Yeah,
and
no, no. As long as we went through, we recorded this part, right? That's helpful because the maybe I'll just summarize by saying that the risk here for is that because we're only doing a pre-IPO raise, let's call it 80 million US, right? And if we get there, I just wanted to assess how safe we are. Because in a sense, right, you could divert all 80 million to completing this phase 2B/3 and just hold the DMD CM, right? So even if they ask for 150 patients, we'll probably be fine. Correct?
yep,
Yeah.
exactly.
So yeah. No, that's very helpful because once we get that, when we go to the public markets, the IPO is actually icing on the cake. It's not a life or death. So the company is already de-risked. That's the most important message that we're trying to get at. And in the course of the due diligence, right? Because the two of you are so involved in the regulatory, you guys have the full picture. But for us, it's like we didn't think 80 was enough at all, right? That was the risk. Yeah,
Got it. Got it. Yep.
yeah,
Exactly.
yeah. Then yeah, I generated two sets of AI research, right, about AskBio and the space, as well as on a device approach, so a medical medtech procedure approach to this called they call it pulmonary ablation, right, on at 3 points. So yeah, when Roger has a chance, right, can you give us your thoughts on those, right? Those are very detailed reports actually. So I'm not saying that they are replacement of us because it's difficult, right? We're going after like first in class indications, right? I'm just saying that could they, because of their discussion with the FDA, raise the bar for us? Yeah, that is the other risk. It may be good or bad, right? Yeah,
But they're not
those
doing HFpEF. I mean, how can they raise the bar for us? I mean, if they're not doing HFpEF, how could they?
the funny thing is, at least for Palnovo, right? They're saying that the HFpEF, they're going to show results for HFpEF patients within a subset of that 500, and they are pretty confident it's going to help dramatically. So by maybe Roger can jump in, maybe Ronald can jump in, right? By blazing on 3 points, the relieving the pressure on that, the HFpEF patients starts. I'm just repeating what it said. I think something like on the 6 minute walk time, like gaining 80 up to 60 to 80 steps more for even HFpEF patients, but doesn't tell me which grade of it. I don't know how they can get an FDA maybe I'm gonna guess, right, like a cross label, right, off label use of it. But in case that affects our bar, yeah, they're just starting, and the US trial also run by Cuvian. That's the interesting thing. So we could ask more about Kings. I only just got this information last Friday. Yeah. Yeah. Anyway, cool. If Ronald can share the two research reports with Roger, right? We can have a follow up discussion eventually, right? On this, it's just something to keep an eye on the horizon. Yeah.
Sure. Yep. I mean, as I
Yeah.
said, so
No.
if it's going to work out for them, we don't know the full details yet, so we should take a look and then comment. But even if it's true, it's going to be just a subset of a very big patient population, and we on the just superficially, you know, my guess is that the patients are not as sick like the ones that we're looking at
Yes,
class.
yes. Most probably the
Um,
case. And they are saying because when I asked them the same question, right, we're researching in a different direction, they are up against Merck's sotatercept, which actually had another clinical win last night in the phase 2, right? They're saying that it's a fully complementary drug. So it's a complementary procedure, and then the drug is 200,000 US dollars. The procedure is 22,000, right? 110. Yeah. So we're learning all this, right? The other thing
yeah.
is when you want them to succeed because they are actually your IPO comp. They go to 2 billion US, then you have a lot easier to explain to the market, right? That's a lucky thing for us. It's just that we needed to figure out the story in case that there are questions from Morgan Stanley, etc. Right? Yeah.
I think you're right. We are not necessarily competitors, right? If anything, they are complementary. And even the Merck
Okay.
program, they're
Yeah.
relieving the pulmonary pressure and therefore improving kind of like the cardiac function, right? But you're not really fixing the heart, and we are targeting the molecular pathway to repair the myocardium, right? So that's a key difference. So if
Yeah.
anything, I would say that they are complementary, and we are dealing with sicker patients too, and they can
Yeah.
be a supplement to our program after gene therapy, perhaps.
Yeah. No, for both Ron and Roger, right? The problem is most of the investors in Hong Kong, they are face value guys, right? They don't have any competitors that they can or comps they can relate to. They're not familiar with sotatercept in the US. By the way, it was a 10 billion US buyout by Merck, right? If they're not familiar with it, they will view these as competitors. They don't realize the degree. That was actually the problem I had when I myself ran into the first draft of the due diligence. I didn't know whether they would raise the bar for us. They would eat in our pie, or they would benchmark our efficacy. So after that, I fully understand. So you two are the deep experts, even in the clinical front, right? The most investors are not. So you probably have to lay that out cleanly and say, hey, we're not going after symptomatic. We're disease modifying at the root of the disease, right? Yeah.
So we're counting on you to come up with a good equity story.
Yeah, yeah. I'm trying to get the story right myself, so now we can get it correctly. But it's awesome. So far, it's really really very very exciting for us. I think myself and my two CIOs, right, and CEO, really love the story. So yeah, we'll definitely do what we can. And later at the investor meeting, right, for the generalists, I will try to put that into the clean format for them, right? It's because they're gonna ask about ask. They're going to clean that up, and then I'll refer to sotatercept and the Argo deal. Yeah.
And Johnson, so strategically, I think it's easier for you to say than for us to say in that case. So
Yes.
happy,
Yes.
you know what I mean, right? So from the
Oh,
marketing
oh, in
perspective,
in in the in the comparables, right, on transaction comp side, absolutely leave it to me. Yeah.
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 And one quick question, right? So shall I meet you at Mandarin Oriental? Because the schedule that you sent me, it doesn't say the locations about these
Yes.
meetings.
Yes. Yes. It is Mandarin rental.
Okay, I'll
Yeah.
meet you
In
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 lot
in, in in fact, I
10:45
don't
or
even
something.
have that. Yeah, I don't even have that location myself. I will go ask him for it.
All right,
Okay.
I'll see you in the
Thanks very
lobby.
much. Thank you both. All right. Thank you.
Thanks,
Happy
BJ.
to meet Roger. Thank you. Take care, guys. Yeah.
Take care. Good luck. Good luck, guys.
Yeah. Hope to see you in person soon. Thank you.